It's good to see you again. I saw you. We talked a couple of years ago, and it was a great bit of information, and honored to have you here. Would you be so kind as to just kind of let folks know your background real quick for the people that aren't familiar with your work?
Yeah, Tom Seyfried. I'm a professor of biology at Boston College here in Chestnut Hill, Massachusetts. And I teach general biology to non-majors and also a class on cancer metabolism to majors in biology and biochemistry. And that's pretty much what I do. And I do a lot of research, basic research on developing new therapies for managing cancer using diet-drug combinations for the non-toxic management of cancer.
Now, you've mentioned, you said that cancer is a mitochondrial metabolic disease. Is that still the position that you hold at this point? Has anything changed in that sort of thought, or is that still pretty clearly what's going on?
Yeah, that's perfectly clear. We just published the paper. It just came out in the online journal Metabolites. That clearly defines cancer as a mitochondrial metabolic disease and not as a genetic disease. So everybody is welcome to read that paper. I'd be happy to address any questions that they might have. My other colleague that was my co-author was Dr. Christos Xenopoulos from the Semmelweis University in Budapest, Hungary. World leader on mitochondrial energy metabolism.
One of the things that you mentioned, before, the thought is that somatic mutations in the DNA. This is kind of the maybe prevailing thought. I don't know. Is it still the prevailing thought that there's these somatic DNA issues? One hit, two hit theory, and then it becomes cancerous. Is that still the sort of the operating principle behind the way we treat cancer today, based on that?
Yeah, it is. Unfortunately, it is exactly that. If you go to the National Cancer Institute website, NCI website, they'll define cancer as a genetic disease. Very complicated. Cancer is a very complicated genetic disease, actually consisting of hundreds of diseases, where somatic mutations are largely responsible for the dysregulated cell growth. So you're right, Shawn, this is a dogmatic view. Despite massive evidence suggesting that cannot be the case, the field seems to mindlessly grasp this without looking at the contrary information that doesn't support that concept.
Yeah, and some of the contrary, information. The biggest thing that I learned from you is that these cancer cells are very heterogeneous in their DNA, in their genetics. You have a cancer that doesn't have the same mutation, and it has multiple different DNA profiles. How does that work with it being? How does that line up with the theory that they have, or can it possibly?
Well, I mean, you're right. I mean, it's already been done by many molecular biologists. If you take any kind of a cancer, liver, colon, bladder, breast, and you separate the cells and you grow each cell up and then clone the cells and make a whole bunch of cells, every cell in the tumor has a different constellation of mutations. So one guy's liver cancer or breast cancer or lung cancer has a very different genetic profile from somebody else's. Yet, no matter what cell is in the tumor, no matter what person has the tumor, regardless of where the tumor is, they're all fermenting. So cancer is the common pathophysiological mechanism is simply a fermentation metabolism dependent on glucose and glutamine. Regardless of the different kinds of cells and the different kinds of mutations, and all that stuff is downstream epiphenomena that people seem to focus more on, not realizing that the common problem is a fermentation metabolism, which is quite easy to manage.
Let me ask you this question, because you mentioned glucose and glutamine as being the two primary fuels, and if the cell cancer can't get enough glucose, it'll revert to glutamine. Do we have any kind of threshold numbers? Like I said, I can, my blood glucose can be really, really high depending on what I eat, or it can be really, really low. Is there analogous numbers in the human body? We could say if your blood glucose is below a certain level, it's less likely to contribute to fuel for cancer. Are we at that level where we can say there's a dose response?
Well, it's a complicated question because it has several different aspects. If you're a healthy person like you are, and many of your followers, blood glucose can rise and fall depending upon your exercise level and what you're eating and things like this, with no impact on your body. Glucose, consistently high glucose levels due to insulin insensitivity associated with type 2 diabetes can lead to systemic inflammation. And systemic inflammation can trigger, over time, damage to respiration. And then the level of glucose, once the cells become damaged and they become more reliant on glucose, then the levels of glucose become important, because it's well documented that if you are diagnosed with any type of tumor, the higher the blood sugar in your body, the faster the tumor will use it and grow. But if you're healthy, it's a very different issue, because the levels will rise and fall with adequate insulin sensitivity. It's only when you are diagnosed with the neoplasm that levels of glucose in your body become important to be monitoring that.
Since I last talked to you, there's been, obviously a lot of research that's occurred in the last 2-3 years. Has anything significantly occurred that you've seen either in human or animal studies that gives further knowledge or supports what we're doing or what you're trying to do with the metabolic therapies?
Yeah, well, this, as you know, I think you interviewed our patient, Pablo Kelly, and he's still alive now, over 7 years with his glioblastoma. It's not cured because it's still there, but he has learned through his diet manipulations to kind of keep it in check. He's managing it. There are a large number of other patients that unfortunately we haven't written up for publication, where they're experiencing very similar outcomes. They're progression-free. I'm not saying any of this, I don't know if we've known 100% sure if anyone's tumor has been completely eradicated, but they're certainly living with it, and they have a very high quality of life. And as long as the tumor can remain either non-detectable or minimally detectable, or not a problem, there's a lot of those people. The problem is it takes a lot of energy on my part to write it up, and because you have to document the histology and all of the details that go into the appropriate diagnosis, and then you have to show the evidence that this tumor is managed, and you have to use various non-invasive technologies, MRI, PET, CAT scans, and these kinds of things. So in other words, to document the benefit of metabolic therapy for managing cancer. The treatment hasn't really been utilized long term. Like someone can say, well, okay, my cancer, like Pablo, his cancer is under management. Okay, if Pablo's here in 25 years and his cancer is still managed, but it's there, is he cured or not? We don't know. But he certainly outlived 95% of anyone else that would have that kind of a tumor. So, and I think we're seeing that now for advanced lung cancer, colon cancer, breast cancer. We're seeing longer and longer term survivors, and people who didn't have to suffer from the toxic adverse effects of more aggressive therapies like immunotherapies and chemotherapy, radiation therapy. These kinds of things do a real damage to your body. And if you can manage the cancer effectively and keep patients in a high state quality of life and still keep that tumor under control, then I consider that a great success.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the general scientific concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
What is, I mean, just to put it in perspective, what is the success rate, and what are some of the comorbidities associated with the current treatment paradigm? We'll describe the current treatment paradigm and how successful is it, and what are some of the problems you see with it?
Well, I mean, we can know that just by looking at the number of people who die each day. Okay, so over 650,000 people a year are dying from cancer, and over 1,600 people a day dying from cancer every day. So how would you measure the success of a disease that kills significant numbers of people, not only in the United States, but throughout the world? In China, it's over 8,000 people a day dying from cancer. And the numbers keep growing. They get worse and worse every year, despite the hype around all these new therapies that, you hear them advertised on TV, Keytruda, Opdivo, and all these immunotherapy, PD-L1, and all this kind of stuff. Yet we still have it's not dropping the death rate. So and the adverse effects, I mean, I'm not saying we have millions of cancer survivors who have taken standards of care, but their bodies have been beat up significantly. I mean, who's going to get healthy from high doses of radiation and toxic chemicals? I mean, in the long term, the body is going to pay a price for that if you survive. Because a lot of people, as I said, 1,600 people a day are not surviving. They're dying from their disease. So I would say that it's a tragedy of monumental proportions, where we've had this disease in our society for such a long time with no major advances in 50, 60 years, despite all the hype you hear about all these new drugs and things. Just look at the American Cancer Society quantifies the number of dead people from cancer every year, from each of the individual types of cancer. So you have this about 43 to 44,000 die from breast cancer every year. You got pancreatic, you got lung cancer, colon cancer, bladder cancer. What's going on? Why we hear all this? Let's raise money for cancer research. What kind of research are you doing if the more money you raise for cancer research, the more cancer you get? So, I mean, there's a real disconnect in this whole cancer thing. And there's no accountability for all the money and all the efforts that are being expended for managing cancer. There's no accountability. There's no outcry in the part of the population say, what the hell are you doing with this disease? How come so many people are suffering immensely and dying? Why would you take a drug that makes your hair fall out, your lips bleeding and killing your microbiome? Why do you do that? You don't have to do that if you understand the biology of the disease. It's a metabolic disease. It can't live without glucose and glutamine. Target glucose and glutamine. You don't have to lose your hair. You don't have to have your gums bleed. You don't have to have all of these adverse effects that we're currently experiencing from a lack of knowledge or a failure to understand the real biological problem of this disease.
Dr. Seyfried, let me ask you just a scenario. Breast cancer, for instance, extremely common. I think one out of 3 or 2 women will be faced with it or something in their lifetime. Traditionally you pick it up on a mammography. There may be a spot on the mammogram. So the question is, we're going to do a needle biopsy, or maybe we're going to go and do a lumpectomy, followed by axillary node dissection, followed by perhaps prophylactic mastectomy or a radiation chemotherapy. How should that be handled from the beginning? If you have a woman that has a small irregularity on a suspicious lesion on a mammography, how should that be handled?
Well, I mean, the way it could be handled, it's not that it would be, but the way it could be handled is that we would consider non-invasive technology to assess the location and size of the mass. Not doing needle biopsy only because it would disturb the microenvironment. And there's significant numbers, and we published a major paper on how many of these needle biopsies can actually spread the tumor. So if the tumor is benign, why would you want to do a needle biopsy on it? If the tumor is malignant, you would never want to do a needle biopsy on it. So you can look at blood work. There's what we call liquid biopsies that are actually now being developed, where you can look at biomarkers in the patient's blood for signals that might indicate a potential malignancy. And if you don't see the signals, it might be benign. I mean, there is false positives and false negatives, of course, like any assessment, whether that's a needle biopsy or not. But the question is, okay, after you would identify it with non-invasive methods, then you would apply metabolic therapy. That is diet drug combinations that would target and kill the tumor cells. If the tumor shrinks down, then one could always do a lumpectomy after the tumor would be reduced in size, because it would be now, if it were an angry or an aggressive tumor, it would be shrunken down in size. You wouldn't allow those cells to metastasize in the first place. So you could probably get the whole thing out. So that, and if it stays pretty much stable, then the patient has considers another lumpectomy, because once you have shrunken down the microenvironment, once you reduce the inflammation in the microenvironment of the mass, it tends to shrivel, becomes reduced in size, and becomes far less aggressive anyway, whatever it was. And then the options become complete removal of the mass, and sometimes the mass goes away completely. It's just like what happened. So you don't really have to do anything. Mastectomy, in my mind, should never be used, only because of a number of scientific publications have shown that it provides no benefit to the patient. Lumpectomy, mastectomy, no difference in overall survival. So why would anyone want to do that in the first place? So there's a lot of strategies that are non-toxic, but it requires participation on the part of the individual. So the individual would have to take it upon themselves to bring themselves into a state of therapeutic ketosis. And while they're in that state, they would be taking certain kinds of drugs that work together with the diet that would restrict glucose and glutamine availability to the mass. And then it either goes away, becomes very small, and then you have different options to finalize the process.
Yeah, because I know last time we talked, glutamine was a challenge to deal with. It's endogenously produced in the body, but so is glucose, to be fair. I mean, we make glucose regardless. There's no one with zero glucose, no matter how many carbohydrates we eat. But what are these drugs that you use? And then I know in the past you talked about something called a press pulse procedure where hyperbaric oxygen was also part of this deal. Is that still part of the program?
Yeah, it can be for certain cancers. But getting back to the glucose issue, on the study by Genetics, where he took 21 obese patients and water-only fasting for I think it was 17 patients, water-only fasting for 21 days, where the ketone levels went way up and the glucose was down around 50 to 60 milligrams per deciliter. Then he injected each of these individuals with a high dose of insulin, which brought the blood sugars down to 0.9 milligrams per deciliter, 0.5 millimolar, which is almost no glucose in the body. And there was no cognitive impairment on any of these patients because their ketone levels were so high. And when you bring blood sugars down to that level, normal cells in the body now compete directly with the cancer cells for the fleetingly small amount of glucose that you would have. So it is possible to bring glucose way, way down, starving the hell out of these tumor cells. Now, the glutamine issue, of course, as you clearly mentioned, glutamine is the most abundant amino acid in the serum in our bloodstream. It's used for the urea cycle. It's used in our immune system. It's used for everything. There's no dietary therapy that I know of that can reduce glutamine. Drugs are needed. And that's where the pulse comes in, because if you take a drug that targets glutamine too aggressively, you can harm your immune system. You can create all kinds of metabolic havoc. So you need to pulse that. Once the patient is in therapeutic ketosis or nutritional ketosis, you can pulse lower doses of drugs that specifically target the glutamine availability to the tumor without harming the normal cells because they've transitioned already over to ketones. It's a beautiful, elegant, simple, non-toxic strategy, putting the maximal pressure on the tumor cells while minimizing any toxicity to the normal cells. The only thing you have to know is how to do it, when to do it, dosage, timing, and scheduling, and that's what we're working on right now.
Yeah, and this may be outside of your purview, but there's somebody asking about exercise with oxygen on board. Is there any benefit to that? I mean, exercise seems to be beneficial for most diseases. I assume it has a benefit in cancer. Is there any evidence on that being effective?
Getting back to the hyperbaric oxygen that I've worked out with my colleague Dom D'Agostino. Once your body is in nutritional ketosis, hyperbaric oxygen seems to really have a positive effect on the growth of the tumor. It seems to reduce the inflammation around the tumor and also creates oxidative stress. So oxidative stress will kill tumor cells, especially when their fuels for fermentation are under restriction. So hyperbaric oxygen can kill tumor cells in a similar way that radiation can kill tumor cells, but without doing any collateral damage to normal cells like radiation can do. Again, there's so many nifty, elegant ways to kill tumor cells without harming the body, and yet making the body even more energy efficient and healthy while you're eliminating the tumor. So you don't have to worry about all this collateral damage. And yes, exercise is absolutely an important component for the overall program. So if you're targeting glucose and glutamine while elevation of ketones, yes, exercise. Some people ask me, should I do high intensity exercise or whatever? But sometimes high intensity exercise can increase systemic inflammation. And you don't want to do anything to increase systemic. Now, a person who's in really good shape might be able to do a little bit higher exercise than some guy who was a former couch potato. So you have to have some level of an exercise program that I think works really well with the drug diet interaction to manage the tumor.
Yeah, I mean, most of the tumors that I've read about, the advanced glioblastomas and things like that have utilized this metabolic therapy. Is there much evidence in some of these smaller, less worrisome tumors, skin cancers and early stage breast cancers and early stage prostate cancer? Have we got any evidence with this sort of treatment with those types of things before it's at the end of kind of at the end of the road?
Well, we don't have as much. I can't really answer that question too much because most of the people that I see are usually stage 4 guys, and whatever the cancer might be, lung, breast, colon. I mean, these people are oftentimes in really, really bad shape. Now, whether someone who would be diagnosed with a minor lesion of some sort, because a lot of times they go into more aggressive lesions. But I think, sure, if you can catch something early, I think they always say, oh, if we catch it early enough, we can do something about it. In my mind, you can't, it's very hard to get cancer if your mitochondria are healthy. Diet, lifestyle issues keep your mitochondria healthy. And if a mitochondria are healthy, it becomes difficult to get cancer in the first place. So, and small lesions and things like this, I think, as we move further and further into this new realm of treating cancer as a metabolic disease, we'll have more data to assess or answer the question that you just posed.
Let me ask you a question, Dr. Seyfried. Obesity is significantly linked to a whole plethora of cancers. Why do you think that is? What is going on metabolically? Is it the same stuff, or what's going on with obesity?
Well, many obese people have an elevated level of systemic inflammation, and that can be picked up by just measuring C-reactive protein. So if they have type 2 diabetes or they have elevations of C-reactive protein, this indicates a state of global inflammation. Inflammation is a powerful risk factor for the development of neoplasia, dysregulated cell growth. So systemic inflammation caused by obesity, because there are some people, I don't want to put all obese people in the same boat, because there are some obese people that have pristine blood work. They have no inflammation. They just happen to carry a lot of extra weight, but their blood work would tell them that they're quite healthy. But on the other hand, there's more obese people that have systemic inflammation in the body, and that can put certain cells in a different organ system at risk for damaging oxidative phosphorylation, which is the initial step in the origin of all cancers, the damage to the oxidative phosphorylation, which Otto Warburg first identified. And then the cells gradually shift and slowly go into a fermentation metabolism, which then leads to the dysregulated cell growth. So as long as you can keep your body healthy and keep inflammation at a minimal, you reduce risk. Let's put it that way. You reduce risk.
Are there any in the United States or perhaps other places in the world, or any places right now currently employing what you're describing for human patients? How possible is it to get metabolic therapy? I mean, obviously we can all go on a diet, but I mean, access to the hyperbaric, access to the drugs you're talking about. Any places that people are going that are doing this currently?
Well, our colleagues in Istanbul, Turkey, I would have to say that this is a work in progress. I think first you have to have a staff of people that actually know what to do and how to do it, and they have to know the concepts. The patient also has to know the concepts. I don't think there are any major clinics in any of them, in the major hospitals. I think it's going to happen in small clinics initially, where they are eager to try and know how to do this. And we're in the process of working up protocols for treating cancer as a metabolic disease. And this will provide a framework and guidelines for clinics that can implement this. There's tremendous interest, to be honest with you, all around the world to do this. There are many physicians that are simply fed up with this crazy toxic approach to cancer. And they really want the knowledge because they're anxious to do this. The question is, how do they do it? What are the dosage, timing, and scheduling issues? Because this is an alternative medicine. When you say a diet, people say, oh, what is a diet? No, this diet is a medicine designed to work with other medicines and drugs to put synergistically attack the tumor from all angles. And we're working out the details right now. What is the best drug? What is the best dosage for that drug? What is the timing for that drug? The patient has to be in therapeutic ketosis before the drug has its maximum effect. We would like to share that knowledge and those processes with others so that they can start treating patients with this. Now, there are clinical trials for glioblastoma, but the patient is irradiated and treated with toxic chemicals before they do metabolic, which is insane. So you don't take someone's precious brain and irradiate it and liquefy it before you start a metabolic therapy. But yet in the main hospitals, they will not presently allow metabolic therapy to be used as an alternative to toxic drugs and radiation. So we have that impediment standing in our way. So simply the institutional review boards will not allow, from all the individuals that I've spoken to, they will not allow metabolic therapy to be used as a first line. Only after toxic radiation and toxic chemicals are shown to fail, then we might want to consider metabolic therapy. So the metabolic therapy now must try to correct the incredible damage to someone's brain done by radiation, as well as poisoning them with drugs that increase inflammation and create all kinds of metabolic havoc. This is absurd. So you want to use metabolic therapy as a first line of attack, and maybe at the very final stage, you might want to use a slight bit of a chemical or a radiation technique, or just to kind of finish off any survivors, but not as a first line. So the smaller clinics would have to be the ones to establish first line use of metabolic therapy. We have to publish those papers. This is what we do. We get the data from the individuals, from the physicians, and we publish the papers like we did for Pablo Kelly. and others that we're working on currently. So yeah, so this is what I've outlined here is the challenges and what we need to do to move forward on this whole thing.
You mentioned therapeutic ketosis. There's a concept of nutritional ketosis, which I think Stephen Finney and Jeff Follick have defined as 0.5 millimolar. What do you mean by therapeutic ketosis? How much ketones do you have to make?
Yeah, this is
glucose.
You're right. You're right. This is still a work in progress, but Dominic D'Agostino and we've spoken about this. It's nutritional ketosis. The question is what level, and we use the glucose ketone index calculator to give us an estimate of when a patient is in nutritional ketosis or therapeutic ketosis. I like to think ketone levels of 2 to 4 millimolar with blood sugar levels about 3.5 millimolar. Then you'll get a GKI value of 2.0 or below. Now, young, healthy people, guys that don't have cancer or any other kinds of problems, can easily get into these zones. We've noticed that many cancer patients, because they also have comorbidities and a variety of other things, have a much greater difficulty. So they're on medications, they're on all kinds of things that interfere with their ability to get into nutritional or therapeutic ketosis. So the patient must be monitored up front, and before we treat them with anything, they've got to get into the nutritional ketosis state before drugs or anything else further is applied. And this could vary from one individual to another, the age of the individual, so and the type of diets that would be facilitatory to getting a patient into a particular ketotic state may differ from one person to the next. So the issue here is get the patient into ketosis first, and then apply drugs that work synergistically with this new physiological state.
Is there any dietary modifications that have an impact on glutamine in a significant way that can be implemented that you can think of?
No, I haven't seen anything yet, because glutamine can be produced from other molecules, other amino acids can be produced from sugar. That's why they call it a non-essential amino acid, because you can make it from other things, and you get it from the diet. But the cancer cell, absolutely, for the cancer cell, it's an essential amino acid because it can't make ATP without glutamine. And without ATP, you can't live. So the glutamine is the gold for the energy for the cancer cell. So we have to use a drug to target that. But the drugs that we use are minimally toxic, especially when the patient is in nutritional ketosis. So that makes it, otherwise you run the risk of harming the patient if they're not in keto, and you will not get the power of the therapeutic benefit if the patient is not in ketosis. So these are the trade-offs.
What is I was going to ask, there's people asking about glutamine and how does a drug interfere with it? Does it interfere with uptake by the cell? How do the drugs work?
Well, it depends on the glutamine drug that you're using. We use 6-deoxynorlucine, which is called DON, and this is a glutamine analog that blocks the ability of, when glutamine is metabolized to glutamate in this glutaminolysis pathway. DON blocks a range of glutaminases. So you don't break, glutamine cannot be used to make ATP. So consequently, you're killing the tumor cell. There are other glutamine analogs that are slowly appearing in the pharmaceutical realm. I have not tested them, and I can't really speak to them, but maybe for blood cancers, you could possibly consider phenylbutyrate. Human body breaks phenylbutyrate down to phenylacetate, and phenylacetate combines with glutamine, and you eliminate it in the urine. And that drug was developed for kids with amino acid disorders and things like this. I don't know if that would be effective against solid tumors, but certainly DON is. And I think there's a couple of others out there. I just haven't had a chance to test them yet. It takes a lot of time and money to test all these different kinds of drugs. But I think that every one of them will work better when the patient is in ketosis, whatever the drug happens to be.
There reportedly are cancers, or at least maybe you can correct me if I'm wrong, but that supposedly thrive on ketones. Is there any validity to that, or where is that coming from?
It's coming from people who don't understand biochemistry or physiology. And it's like he said, she said, they just don't understand. I published a big paper with Christos and some of my colleagues, where I combed the literature looking at the structure and function of mitochondria from patient tissues for all the major cancers that we know of, bladder, colon, breast, all of the major cancers. And we look at the structure and function of the mitochondria in those tissues. And in every one of those major cancer, there was evidence of abnormalities in the structure and function of the mitochondria in those tissues. You cannot, ketones are a non-fermentable fuel. Ketone bodies cannot be used for energy in cells that have abnormalities in the number, structure, and function of the mitochondria. So people simply ignore the evidence that says that ketones cannot. If you take glucose and glutamine away, ketones cannot replace either glucose or glutamine to give the tumor a level of advantage, or survival advantage, I should say. And if ketones can get up in the 10 millimolar range, which is a little bit high, but not ketoacidosis, which comes in at about 15 to 20 millimolar, 8 to 9 millimolar can actually be toxic to tumor cells. This is an area that we don't like to push patients that far into ketosis, but certainly ketones cannot be used. Now, what's interesting, fatty acids actually are a turbocharger for the cancer cells to use glucose and glutamine. So when people say, oh yeah, some cancer cells thrive on fatty acids, no, they don't. They can't be fermented either. But fatty acids act like a turbocharger for letting the tumor cells use glucose and glutamine. So they think the fatty acids are doing a big, if you have glucose and glutamine in the tumor microenvironment, fatty acids could make them grow faster. However, if you take away glucose and glutamine, the fatty acids cannot replace either glucose and glutamine, just like ketones. So when people say, oh, the tumor cells are using keto, they're not. They would have to deny a fundamental tenet of the field of biology that structure determines function. It's an evolutionarily conserved concept. So obviously people, they may not understand evolution, they may not understand structure and function relationships in organelles, and therefore they would come out with insane statements that say they can use ketones. So, it's just, sometimes I get so frustrated. They don't read the scientific literature, and if they do, they don't understand it. So cancers are dependent on, cancers can live in cyanide and without oxygen. So obviously they're not using oxidative phosphorylation. You need oxidative phosphorylation. Fatty acids and ketone bodies generate energy through oxidative phosphorylation. Cancer cells don't use oxidative phosphorylation. Therefore, ketone bodies and fatty acids cannot be used as a direct energetic fuel for cancer cells. Period.
Question on, if we're eating, assuming, to lower glucose, most people are going to choose some sort of low carbohydrate diet. The question then becomes protein, fat. Is there a concern around perhaps too much protein driving glutamate or driving, too much gluconeogenesis, therefore leading to higher glucose levels? Is that something that is impacted by the diet?
Yeah, well, that's an extremely important question. And that's an individual. Patients have to look at themselves, and if they know their GKI values, glucose ketone index, like Pablo went on a carnivore diet, and he was able to maintain a lower GKI. I think there are vegans who can choose the right plants or whatever they want to choose, and they could possibly get into a similar state. Everything that we've tested, when you eat too much of anything, if you're eating too much plant material, or if you eat too much meat, you may throw yourself out of ketosis for various reasons. So I think everything has to be put into the balance with that individual's own biochemistry and physiology. So I think a Paleolithic carnivore diet could be just as effective as a plant-based diet, as long as the patient is in the state of nutritional or therapeutic ketosis. How that individual gets into that state, how much fat or protein or whatever they're eating. So this is what makes it interesting, because what may put me into ketosis may not be the same as what puts you into ketosis, but yet we know that we need to get there in order to have the targeting of glucose and glutamine as efficiently as we can get it. So I think that's more of an individual question, but it would be extremely interesting as we go forward to see what diets are the most effective. All I can say is that a diet that would be low glycemic, low in carbohydrates should be better to get into ketosis than a diet high in carbohydrates. Just makes sense.
Yeah, that's pretty clear. Let me ask you, Dr. Seyfried, with the relatively kind of abysmal results we're getting with the standard of care with cancer, the morbidity, the lack of survival rates, the real lack of meaningful impact. What is sort of preventing stuff like your techniques being utilized in a wider fashion? Is there a pushback from pharmaceutical companies? What's driving the standard of care to be so ineffective?
Well, it's revenue, clearly. It's revenue generation over patient outcome. There's no question about that. I mean, the science tells us that it's a mitochondrial metabolic disease. If that's the evidence is overwhelming, the disease can be modified, managed very, very quickly. So then you go to the oncology clinics and you ask, why are you not just the very same question? Well, I never heard of I never heard of it. Do you know who Otto Warburg is? No, I never heard of him. Well, how in the hell can you manage somebody's cancer if you don't even know the basic biology of the disease that you're managing, right? So, and the other thing too is hospitals have to generate significant revenue. The business model for a metabolic approach to cancer has not yet been developed. In other words, there has to, if you're going to have a management for cancer, it has to generate enough revenue. If you want to replace radiation and toxic chemicals, you have to figure out another business plan by which it's doing this. I mean, right now, I'm more interested in patient outcome than in revenue generation. I mean, I'm not opposed to people trying to make money on a new approach to cancer management that doesn't cause toxicity. But I think that doing what we're doing today to these people for the prime reason of generating revenue for hospitals, physicians, and pharmaceutical industries is the public has to come up with a statement about this. I mean, when you say, oh, we have, let's raise money for breast cancer. The more money we raise for breast cancer, the more breast cancer you get. What are they doing with all the money that we're raising? And if breast cancer metabolically is no different than colon cancer, bladder cancer, or whatever, it's all the same disease. It's one disease having a common metabolic problem. So march on Washington. Tell President Biden that, hey, we got to do something about this cancer because it's an easily manageable disease. It's just that the people at the hierarchy doesn't want to do it that way, right? So my job is just to show how easy it is to manage the disease and provide the basic concepts that underlie this. Yeah, revenue generation. Say it again. It's revenue generation.
I'm truly shocked that the pharmaceutical companies are trying to make money or the hospitals are trying to make money.
Yeah, it's unbelievable they would do something like that, right?
And I guess I'd be remiss not to mention, we're in the midst of this pandemic. There has been maybe $1 trillion spent or more in messaging and this sort of stuff. And to be fair, I mean, heart disease and cancer kill as many as this pandemic has every single year over and over again. And where is the money for that? Why don't we have money for these types of things, these chronic diseases, which in your estimation and in my estimation are better managed than what we do now?
Well, it's all diet and lifestyle issues. But the food industry produces poorly nutritious foods that make us sick, and the pharmaceutical industry produces the drugs to try to make us healthy, right? It's like a big vicious cycle. But people have choices. They try to do the best they can under the environment that we're in, and these are difficult questions, difficult situations. But I think there is a clear strategy. There is a clear path forward. The question is, the patients, individuals are embracing this, but there'll be tremendous resistance on the part of the various industrial links to this. Academics as well. We have tens of thousands of scientific research grants that are going to study gene mutations and all these kinds of things in these various diseases. I think, they're all linked to a diet and lifestyle. You get diabetes, Alzheimer's disease, various chronic diseases, cardiovascular disease. It's all linked in one way or another to abnormalities in diets and lifestyles. And in my recent paper, I said with great apes, the chimpanzees, they're the closest biological relative that we have, 98% similar to us in gene and protein sequence. And there's never been a documented case of breast cancer in a chimpanzee. What the hell's going on with that? Well, they're eating the same food that today, whether they're in the zoo or in the wild, that they ate when they first evolved as a species. But we are not doing that. Let's put chimpanzees on the American diet and see what happens to their. They'll probably be very happy as they're slowly dying, right? So, yeah, these are complicated questions, but there are answers, but there are also choices that can stand in the way of the right answer.
That's fascinating. I didn't know that primates don't develop breast cancer outside of humans. It's really interesting. Let me ask you just a hypothetical. Let's say you're, some 47-year-old guy in Texas, and you're diagnosed with a 3-centimeter melanoma on your forearm. How do we manage that?
Yeah. Well, that's that. I would say, because we know what melanoma, malignant melanoma, comes from the melanocyte. And the melanocyte is a derivative of the immune system and the macrophage class. It's a resident macrophage of the skin. We know from the work of the Newscombs in England that glutamine is the prime fuel for macrophages of the immune system. So it would be clear to me to initiate, get the glucose down, get the patient into therapeutic ketosis, and especially use glutamine targeting drugs on that kind of tumor. Because as a matter of fact, most metastatic, our data has shown, and others have shown, that most metastatic cancers, whether it's a melanoma or triple negative breast or lung, they're all of a macrophage. That's why they metastasize. They grow so rapidly because they have so much access to glutamine, and no one's targeting the glutamine. So the outcome for these aggressive cancers, people dying, should not be surprising, because you're not targeting the very fuel that they need to grow. So once people understand the biology, this cancer is going to be a relatively easy disease to manage. People say, well, I can't believe you. This is a complicated. Because they don't. If you understand the biology, then it becomes, oh my God, you can manage this disease not only on the cheap but without toxicity. So I think that's the hardest thing for people to come to realize, especially if you go and talk to the so-called experts. Who the hell are they experts when you have 1,600 people a day dying? I would say that there aren't many of them out there. You're going to get a lot of pushback. But again, melanoma is a glutamine a cell that depends on glutamine. Let's target the glutamine. They're all going to be the same. They're all going to respond similarly. So this is the way it is. I think I don't know how many years it's going to take for people to come to know this, but the path we're on right now is not successful.
Are there cancers that you mentioned melanoma being derived from, basically a cell line of macrophages. Are there cells that will utilize glutamine preferentially over glucose? That is to say, let me just suppose that most of us have some early cancer cell, maybe one cell in our body, and we all have a few of them, and we're continually taking care of it through immune surveillance. Are there cells that can, if you keep your glucose low and you're doing the right thing and exercising, are there cells that are going to just, despite all that, just use glutamine and develop? Or do we know that?
Yeah, no, I have it. We have it. We know it. I can take away all the glucose to some of our tumor cells. They can't grow, but they can live. They make the ATP on glutamine. You need the glucose carbons to grow. Glucose allows the cell to make new membranes, new DNA, new RNA, new proteins, the carbons from glucose. But it's the glutamine that allows the cell to make the energy. Now, the cell can stay alive on the energy, but it can't grow because it doesn't have enough glucose. So that means it could grow very, very slowly. It's only when you take away the energy and you take away the fuel for new membranes and new DNA that you get an implosion and kill the cell. But you're absolutely correct. You can lower glucose to very, very low levels, but some of these tumor cells will hang on. and they're hanging on because they have that glutamine to give them the energy. And I also know that when we take those same cancer cells and we grow them in a load of glucose without glutamine, they die. So the glucose by itself is not giving them the energy. So even though they have all the building blocks to grow new tumor, they can't grow because they don't have the energy. You just drop a little glutamine in there, it's like throwing gasoline on the fire. These things explode. So our press pulse therapeutic strategy requires the simultaneous targeting of both glucose and glutamine. Otherwise, it's like closing the front door and the thing runs out the back door, or vice versa. You've got to shut off both sources of fuel and sources for metabolites to grow. That's when you get the success. And right now, I know of no clinic or oncology center doing this. And consequently, you don't get the results that you should get.
Is there any relationship between the level of ketones and the glutamine levels? Is there a relationship between those two things? Because people are asking me about high-fat diets versus low-protein diets. Is there
well,
a relationship?
In the paper we just published with Pablo Kelly, we wrote the mechanism in there. And, it's very interesting. If you can get your ketones high enough, it siphons off some of the coenzyme A to make acetyl-CoA, and therefore blocking that energy pathway, the glutaminolysis pathway. But it's a kind of a slight pressure interference with the glutaminolysis pathway by forcing the body to make ketone bodies using a coenzyme A. You siphon off some of the fuel for the pathway generating ATP. But it's not going to be sufficient in my mind by itself. I think if you just pushed in a little glutamine targeting drug on top of a good therapeutic ketosis, that's when you really get the power of blocking that energetic glutaminolysis pathway. So, but the answer is yes, you can get some effect. It may not be as good or strong as you could if you threw in a little of the glutamine targeting drug.
What if any are the roles of exogenous ketones in cancer therapy? Are there any? Is it work? Does it require something else besides that?
Yeah, that's a really good question. I like to defer that question to my good friend and colleague Dominic D'Agostino, who is actually developing ketone formulations that we think. And my late good friend Richard Veach was developing formulations. It becomes harder to test in the preclinical system because the animals won't eat it because it's bitter. These ketones are bitter. But for humans, of course, you can take it in pill form and you don't have to deal with the horrible taste. And therefore, the therapeutic, the measurements would be probably better in humans. Because the bottom line is how low you can get your glucose and how high you can get your ketones to put, and if you can do it by an oral additive, then that would be certainly a push in the right direction. But I think Dom has some reservations about some of the ketone things that are available on the web. Some of them are not balanced the right way. Some of them are bogus. He has more of an in-depth discussion on the best types of ketones, additive ketone drinks and this kind of thing. So we haven't tested that as much in the mice, but we would like to do that. But we'd have to gavage it. We'd have to bypass the taste issue and then deal with it that way.
Is there any, I guess, I'd seen some of your work, with dogs regressing tumors. What's the most impressive animal stuff you have right now with this? I know you've done a lot of studies in mice and rats. What's the latest stuff in animals that we can look at?
Well, I think Lauren Nations is a veterinarian from Florida. And Lauren presented some really unbelievable results at the last meeting we went to, where he's feeding the dogs that have cancer these keto, zero carbohydrate, like all meat, Paleolithic, with really remarkable, unbelievable results. So I think the dog, and as I said, the paper we published was spectacular, where that dog, Now that Minka dog, the cancer, I talked to the owner. The dog, it was a pit bull that finally died at like 16 years of age from a heart failure, but the cancer never came back. So it outlived its normal lifespan, and the cancer was completely eradicated using ketogenic metabolic therapy. And Dr. Lauren Nations is getting very spectacular results with his veterinary population, with his canine population. I don't know about cats. I haven't heard too much about that. But the vets are starting to use it, as long as they can get over the stigma about the raw meat issue. You know, I told you they don't like to feed. They were worried about some damn salmonella in the meat. I mean, the dogs eat crap and everything. I mean, wouldn't worry too much about that, especially if you're just, you know, cutting their calories and giving them a good, you know, healthy raw meat. Some people grind up whole chickens with the raw chicken with the bone and all the guts, and they package it into patties. And the dogs, you know, they inhale this stuff, and you give it to them in small amounts. And this is their natural food. I mean, all of these imes and all these kibbles and bits and old raw, it's all crap, you know, and it kills the dogs just as much as it kills us. So, you know, you can definitely reduce cancer in dogs by doing the same strategy. All cancers are the same, whether it's a mouse, a dog, a human. They're all dependent on fermentation. It's just unbelievable. They need glucose and glutamine. So the diet and drugs can really play a big role in reducing this.
Yeah, I mean, absolutely. I mean, I have my dogs on a meat-based diet. They do great. They have no issues whatsoever. You know, it seems to make sense. I mean, you think about what a dog is. It's essentially a wolf that's been, you know, bred down a little bit. They should have the same diet, you'd assume.
Yeah, I mean, you're absolutely right.
Dr. Seyfried, unfortunately, we're running out of time. It's been an absolute honor and pleasure to have you here. Where can people go to find out more about either your work or what you're up to, or what do you have any upcoming publications or studies that you have in progress that you'd like to share, or how do people find you?
Yeah, well, I publish most of my stuff open access, so you just have to look up PubMed and look my name up for recent publications, because they're all out there. Anybody who has access to the computer and the web can get these papers. And we receive a significant support for our research through Travis Christofferson's Foundation of Cancer Metabolic Therapies. He wrote the book Tripping Over the Truth, the Rise of the Metabolic thing. So we're very, very gratitude for Travis's organization, and other people are starting to support us through personal donations to the Boston College. And the longer we can develop the preclinical system and define the parameters for treatment, then it just becomes a choice. Do you want toxic therapy, or do you want non-toxic therapy? That becomes a choice. And eventually this is going to be the way cancer is treated. I just don't know how long it's going to take. As you said, there's a lot of barriers and roadblocks in the way, but anyway, yeah, so we're moving ahead as fast as we can with the resources that we have. And I don't know how long it's going to take, but this will be the best way to manage cancer for the majority of people on the planet.
Well, I hope to see that come to fruition, and thank you for continuing to do what you do, and wish you good luck. And hopefully, you know, people out there that want to support you can help do that, because I think we'd all like to see cancer, you know, be minimized, and not where it is now, where, you know, we all have a good chance of getting it as it stands right now. So thank you again for coming on, Dr. Seyfried. Again, it's always been a pleasure talking to you, and good luck with all the research.
Well, thank you very much. It was a pleasure to be here. Thank you.